Muscle contraction starts with an action potential sent down the neuron into the synaptic end bulb. The charge from this action potential builds up until it's enough to open up the voltage gate. When it's open, it allows a channel for calcium ions to travel through into the neuron, and the calcium ions cause the release of acetylcholine through exocytosis into the synaptic cleft of the neuromuscular junction. The acetylcholine floats around this cleft until it finds receptors to bind to. When it binds to the receptors and the ligand gates, it allows the ligand gates to open up and let sodium in. The sodium ion's charge allows an overall positive charge on this side of the membrane, and this overall positive charge builds up until it can open up a voltage gate to the T tubule and release an action potential down the T tubule. And that action potential travels down until it finds another voltage gate with the sarcoplasmic reticulum. And when it gets enough charge, that voltage gate opens and allows calcium ions to come out. And they float around until they find a troponin. And they bind to that troponin, cause the troponin to make the tropomyosin move out of the way of the myosin so they can bind to the actin. Now, myosin and actin can't have a little more to it when it comes to their binding. They need ATP. Or at least ATP is involved. ATP binds to the myosin head, causing it to cock. ATP goes through hydrolysis and becomes ADP when the bond with the third phosphate group breaks. When it becomes ADP, it uncocks and reaches up and grabs onto the actin. The ADP comes off. The myosin head keeps pulling on the actin until it sees another ATP come along, and it prefers ATP, so it'll let go of the actin to bind again to the ATP, recock, go through the whole cycle again, hydrolysis into ADP, uncock when it becomes ADP, let go of ADP and keep pulling. It repeats this cycle and shortening the sarcomere until something tells it to stop. One of the ways to tell it to stop is to stop sending action potentials. Without the action potentials, there's not enough charge for this gate to open. Without this gate, there's no calcium inside. Without calcium, acetylcholine is not released. No acetylcholine release means there's nothing to bind the ligand gates. And sodium cannot pass over into the sarcolemma. Without the charge from the sodium, there's not enough charge to open up the voltage gate. And an action potential is not sent down the T tubule. Without that, there's not enough charge to open the voltage gate for the calcium. And the calcium's not released. Without the calcium, there's nothing to bind the troponin, and troponin does not move the tropomyosin out of the way for the myosin to bind to the actin, and there's no contraction. When you have extra acetylcholine, or you just don't want it to any more contractions, there's 3 ways for it to be gotten rid of. 1, you can just let it diffuse out of the synaptic cleft. 2, you can reabsorb it into the synaptic end bulb. Or 3, you can use an enzyme called acetylcholine esterase and just have that break down the acetylcholine so it can't be used anymore. Sodium leaves the sarcolemma using sodium pumps, and without the charge from the ion, there's nothing to open this voltage gate anymore, and so there's no action potential to open up the voltage gate for the calcium. So the calcium voltage gate closes and no more is released. The extra calcium is eventually pumped out using a calcium active transport pump. And without the calcium, the troponin has nothing to keep it from letting the tropomyosin block the binding sites anymore, so it kicks the myosin out of the way, and the microfilaments spread apart and the muscle relaxes. Now, one of the things that can cause a problem with the muscle fibers is something called botulism. That's caused by a bacteria called Clostridium botulinum, and it blocks the release of acetylcholine. And like I said before, without acetylcholine, there's nothing to bind to the receptors. So then sodium can't cross over into the membrane. Without that charge from the sodium, this gate doesn't open. The action potential doesn't go. There's nothing to open the gate for the calcium. Calcium's not released, so it can't bind to troponin. Without anything to bind to troponin, it won't move the tropomyosin out of the way for the myosin to bind to the actin.